Well, welcome back to the Classics in Immunology Journal Club. And this week we're going to do one of my papers, which I've deemed a classic, and I hope to be able to convince you. But before we get started, don't forget to go on the YouTube channel and like the video and click on the bell and then scroll on down and go to my website and click on the writing tab. And you'll find all the classic papers of immunology there, as well as other the social medias that I contribute, particularly to Twitter. So you might want to follow me over there. So today we are going to do a paper that is entitled "The Interleukin 2 T Cell System: A New Cell Growth Model." It was published in Science in June of 1984. We submitted it in December of 1983. Now, to give you a little background on this, on the people, the authors are Doreen Cantrell and myself. And Doreen was a postdoctoral fellow who had just come to my lab about a year previously and began doing studies on the IL-2 receptor and so forth. And her background was that she graduated from Nottingham University in the UK, and she had spent her entire graduate time working with the flow cytometer. And of course, back in those days, the flow cytometer had essentially supplanted the microscope as the way to do immunological experiments, because not only could you see the cells that would light up with a fluorescent antibody, that's what we were doing with the microscope, but with the flow cytometer, you could analyze individual cells. Number one. Number two, you could enumerate the number of molecules that were fluorescent on the cell. And then ultimately they're able to do the same thing inside the cell and molecules inside the cells. And so it was a quantum leap forward in terms of immunological assays and experiments. And so the good news was that Doreen had done her whole background in the flow cytometer. And just prior to that, we had completed a series of experiments in collaboration with Tom Waldman's lab at the NIH, because Tom had an antibody that one of his postdocs had created, Takashi Uchiyama from Kyoto, that looked like it tracked on the IL-2 receptor because it was only expressed on antigen-activated T cells and so forth. And so we did a series of experiments that we published in 1982 in Nature that essentially pinned down the fact that their antibodies would block radiolabeled IL-2 binding to the receptor. So this was the first antibody that we had that would allow us to use the flow cytometer, and we could then combine quantitative assay of radiolabeled IL-2 binding with the fluorescence, the flow cytometer, looking at a whole population of cells at an individual cell level. So that was good. We had quantitative and qualitative, as I'll get into. In the intro of this paper, introduction, we lay out the problem that we're going to attack in this paper. And the first thing we say is that T cell mitosis results from the interleukin 2 receptor interaction. Frank's statement in the very beginning, and then it's reference to all of our previous papers that date back to 1977 and so forth. And we go on to say that it's possible, because of the advances that have happened in this whole scene, that it's possible to take new approaches to trying to understand what the molecular determinants are of T cell cycle progression. Those developments were the fact that we had made monoclonal antibodies reactive with IL-2 itself, and we could use them to purify IL-2 to homogeneity, and we could demonstrate that, prove that, which is a huge step forward. And as I said, we had both quantitative assays for the IL-2 receptor number, as well as with the flow cytometer. We had a new qualitative assay, so we could look at the whole population of cells, and we didn't have to rely only on the mean number that we got from our radiolabeled binding assay. And we had worked out a way to synchronize T cells because of the year prior to these experiments, Doreen had been analyzing T cell growth in T cell populations and looking at it from the standpoint of their IL-2 receptor number. And we knew that if we took human peripheral blood cells and activated them with PHA or anti-T3 or something of that sort to activate the T cell receptor, that the cells would then express IL-2 receptors, and they'd go up to a peak at about 48, 72 hours. And then if we kept those cells, we could keep them growing in IL-2 over the course of the next couple of weeks. And gradually, the IL-2 receptor number would decline, even though we kept the IL-2 concentration high, so they couldn't have been due to the fact that they weren't making IL-2. And so we used that system to activate them, have them grow up, and then we could then wash them out of the interleukin-2 and rest them for a few days. And then we could re-stimulate their T cell receptor, and they would rapidly re-express the IL-2 receptor. And then we could add IL-2 at a time zero, and then follow the population now that they were synchronized, so that all the cells within the population were in the G0, G1 phase of the cell cycle. We could then add IL-2, and they would then synchronously grow, and we could follow that. That was a good thing. Now, on the basis of the experiments that we did in this paper, we concluded that the T cell cycle progression is predictable. You can predict when cells were going to undergo DNA replication and mitosis because it depended on a critical threshold of IL-2 receptor interactions that had to occur before the cell would then make the decision to divide. And the decision to divide was an all or none decision. As you might surmise, a cell doesn't want to sort of replicate maybe 10 or 20% of its DNA and then say, well, no, I don't think I'll go any further. That wouldn't be very compatible with life in a very general sense. So once it undergoes this critical decision, it's an all or none phenomenon, and everything it fires after that. Now, one of the things that was known in the cell cycle literature, and of course the cell biologists have been studying cell cycle cells undergoing proliferative cycles. They've been studying them for 50 years, going back to the 1930s, essentially, and they studied all kinds of cells, prokaryotes, eukaryotes, yeast, mammalian cells, avian cells, and so forth, human, mouse, rat, whatever, and they were all exactly the same. And that was that the cells, if they took, what they did, they started taking movies, time-lapse cinematography of their cell cultures, and they could then time how long they could look at a cell and they could mark that cell, and then they could say, okay, how long is it going to take to undergo division? And they could then plot the data and so forth. And when they did that, they found, when they plotted the rate of cell growth, cell cycle progression, they found that there was a normal shaped curve when it was plotted on a logarithmic abscissa. So they called that the rate normal distribution of cell cycle times. And that was true of all the cells that anybody had studied for the last 50 years. So a little bit about the history that we went into then in the introduction part of this paper is that, as I said, that the cell cycle times or rates of cell cycle progression were distributed normally about a logarithmic curve. And a bunch of studies have been done to focus down on the which stage of the cell cycle was the most variable, and they found it was all in G1. And so that was really the determining factor. The question was, what determines the variable rate of G1 in a homogeneous cell population? The other thing that was noticed to us is that if you just kept these cells in culture and passed them every day or whenever, you didn't select out for more rapidly growing cells. So whatever was determining the variability of their growth rate, it wasn't genetically determined. And that was very important. And on the basis of this, two models had been suggested. Back in 1962, a group essentially came to the conclusion that ultimately became called the deterministic theory of cell cycle progression, and it assumes that the cells in a population, even if they're a cloned population, so that they're genetically identical, it assumes that there are small differences, a whole bunch of small differences in one cell to another cell to another cell, and that accounts for the variability. So that was the deterministic model from 1962. And then the probabilistic model was proposed in 1970. And in contrast, it said that it assumes that cells were identical. And it was just a matter of chance when the cell decided to go or not to go through the cell cycle. On the basis of that, we decided that what you really needed to do was you needed to correlate the kinetics of the growth of the population, which is what everybody had done up until this point in time, with a quantitative examination of the critical molecules involved in telling the cell to grow or to not to grow. And of course, up until this point in time, nobody knew what the critical molecules were. All of the cell systems that have been examined were primarily, particularly in the mammalian and the avian kinds of cells, they were using serum as their growth stimulus. So they could stop a population from undergoing cell cycle progression by removing the serum from the culture medium, and then they could add it back again, and then the cells would grow again. They didn't know what it was in the serum that was causing that. They didn't know whether it was nutritional or whether it was hormonal or whether there were special growth factors or not. We knew at this point in time that the initiation of at least T cell cycle progression was inferred upon the population by the antigen interacting with the antigen, T cell antigen receptor. That set everything in motion. But if you didn't have any IL-2, or if you blocked IL-2, the cells would not undergo cell cycle progression, would not replicate their DNA, and would not undergo mitosis. So the IL-2 is responsible for what the cell cycle people call progression through G1 into DNA, into the S phase, the G2 and M phase, and the whole banana. In the materials and methods in this paper, this is the method that we worked out, that Doreen essentially worked out, and that it became stock for our lab for the next decade or two. We take peripheral blood mononuclear cells from each other in the laboratory. We'd partially purify them over a density gradient with Ficol and HiPEG, and then put them into culture and activate the cells with PHA, phytohemagglutinin. And we would culture the cells then in culture, adding IL-2 for about 10 days. The cells would grow up and to a much larger number of cells. And then we would take the cells, take them out of their IL-2 and put them in fresh medium for 3 days and let them rest. And when they rested, what they would do is that they would all essentially accumulate in the G0 or G1 phase of the cell cycle. And they just sit there. And then we could reactivate. We knew that the IL-2 receptor expression was low at that stage of the game. And so we knew that we could reactivate the expression of IL-2 receptors by adding PHA back again, and we had to add a much lower concentration of PHA. And they would rapidly then re-express the IL-2 receptor. And then we could add IL-2 and they would take off. And that was our system. And so in the results of this paper, and now we should do some screen sharing, this depicts the heterogeneous distribution of IL-2 receptors on a receptor positive population of peripheral blood T cells. And this is a flow cytometry plot. In A, we have a linear scale in terms of receptor fluorescence intensity of the monoclonal antibody to the IL-2 receptor. And on the bottom here in B, we have a logarithmic scale, and you can see they're markedly different. There are some cells that are so high, they're way off scale, and in the linear plot. So the best way to plot this is in a log plot, And when you look at that log plot, you say to yourself, well, that's what is called a normal distribution. Some cells have 1,000-fold higher density of IL-2 receptors than other cells. So you immediately say to yourself, well, I wonder if it makes any difference how many IL-2 receptors you have. And that was critical. That very plot told us a lot, because we already knew that when you plotted the rate of cell growth of a population, didn't matter whether it's T cells or not, you'd come up with the same log normal distribution of the rate of cell cycle progression. So these two things this thing tipped us off. If you then go down into figure 2, it shows the IL-2 concentration dependent T cell cycle progression. And what we've done here now is synchronize the cells by our 10-day, 3-day system of stimulation and growth and then withdrawal of the IL-2. And now we've added back the IL-2 and at intervals thereafter then done 1-hour tritiated thymidine pulses on the cell populations. And the different symbols in this figure are different concentrations of IL-2. And this is at 22 hours after the initiation of the culture over here at time 0. This is a supersaturating concentration of IL-2 at 500 picomolar. This is 100 picomolar. This is 50 picomolar. And this is 25 picomolar. And you can see that the highest concentration of IL-2 is causing these cells to undergo cell cycle progression more rapidly. If you then look at just one time point here and plot that against the logarithmic concentration of IL-2, what you get is a symmetrically sigmoid log dose response curve, which we had already shown in our several other papers, that this was characteristic of an IL-2 dose response curve. The other thing that we could do using the flow cytometer was to stain the nuclei of these cells with propydium iodide, and which would quantitatively stain the DNA. And you can see here is a plot at time 0 and or at very low concentrations of IL-2, and they're all the cells are homogeneous and they're in G0, G1. Whereas if you then look at the other end here, you can see that some of the cells, a lot of them are still in G0, G1, but about half of them have moved off because they're starting to replicate their DNA. And here they've duplicated their DNA in this small peak here. And so we had both a quantitative and a qualitative assay to be able to look at the effect of IL-2 on these cells. So what we concluded on the basis of that is that the proportion of cells entering the cell cycle depends on the IL-2 concentration, as shown in both of these plots. In figure 3, which is here, we wondered whether or not how long the IL-2 had to be with the cells to make them undergo cell cycle progression. And so we did experiments whereby we cycled the cells, synchronized the cells, put them back into culture at varying concentrations of IL-2 again, and at varying time periods. We looked at the thymidine incorporation of these cells in both the inset plot and the plot here. And I think this is the easiest one to look at. If you look at the cells whereby back up a little bit. If we washed out the IL-2 after only 3 hours, none of the cells underwent cell cycle progression. And that's down here in the solid circles. In the open circles, we have cells that we've kept in to IL-2 for 6 hours and then put them back with no IL-2. And some of the cells have started to replicate their DNA. And then at 11 hours, about half of them ultimately at the highest concentration of IL-2 replicated their DNA. But the ones that were left with IL-2 during the whole time period, here and here, And so the symbols are the same in this plot as well as that plot. It looked as though, even though we knew that the binding of IL-2 to the receptor would occur very, very rapidly within seconds and minutes after you would add the IL-2 to the cells, and it would come to steady state within 10 minutes. But yet, if you washed it out after 3 hours, none of the cells would undergo cell cycle progression. And so that was really sort of enlightening on the one hand and strange on the other, because we said to ourselves, well, why is that? It looks as though there has to be the time interval, the duration of the interleukin-2 receptor interaction is necessary to build to some critical threshold of IL-2 receptor interactions before the cells begin to go. And you can increase the number of cycling cells by increasing the exposure time. So then we went on, and in figure 4, down here, we had 2 different populations of T cells that we had assayed for the IL-2 receptor, and one population had a higher number of IL-2 receptors than the other. We synchronized and we put them back into culture, and we then added IL-2 and then followed them over time. And this is the cell population that had a higher number of receptors, and this is the one that had a lower number of receptors. So that the population that had a high number of receptors was faster than the one with the lower number of receptors in terms of entering the cell cycle. So that was interesting. And the next thing we did, we wondered what would happen if we took a population of cells and use the fluorescence activated cell sorter to sort the cells, stain them with the antibody, and then sort them into high and low IL-2 receptor densities. And so this is the table 1 here is the proliferative response of IL-2 receptor positive subpopulations, high and low receptors based on the receptor density. And these are 5 different experiments done over several months. And we looked at the thymidine incorporation after, say, 24 hours. The unseparated population gave, in this first experiment, 10,000 counts per minute. The low receptor was 6,500, and the high density subset was 12,500. And you can look then at all of these experiments, and they all basically are telling you the same thing, that it does make a difference if you've got more receptors. Because if you have more receptors, you are faster than if you have a lower number of receptors. And this is really shown graphically here. Here's are the flow cytometry plots with a linear scale here. This is the unseparated population. Now they're FACS sorted for low receptor density and high receptor density. And here is an experiment where they were placed into culture and then pulsed at different time intervals. And the high receptor density population is in the solid circles. The low receptor density population is in the open circles. And the triangles here is in the unseparated population. And you basically see the same plot with different IL-2 concentrations. So those were the data. And we'll stop sharing here and come back to the full screen. On the basis of that, all of those data that we did, and these experiments were spread over a long time period, at least a year, we realized that the log normal receptor density, the normal distribution on a logarithmic scale, was responsible for the normal sigmoid IL-2 sigmoid dose response curve on the one hand, but it was also responsible for the cell cycle times. The fastest cells were the ones that had the highest density of receptors. And so, I mean, that's all makes sense. And now it sounds sort of trite to be able to say that. But at that time, it was revolutionary. And so this was really a eureka kind of an experiment, a series of experiments in our business. And the conclusions that we come to in this paper, we first say that the IL-2 induced T cell cycle progression is a quantal, and so that's a cell cycle term that means all or none. Either it does it or it doesn't do it. And in cell biology, I mean, the other thing you can think about is the neuromuscular junction. The muscle either twitches or it doesn't twitch. So it's an all or none phenomenon. And it's dependent on only three variables that we could, on the basis of all of our experiments. And those three variables are the IL-2 receptor concentration, the receptor concentration or density on the cell surface, and the duration that the IL-2 receptor interaction has to occur. And so the log normal distribution of the receptors and the density of the receptors gave us a new insight, essentially. And because the previous investigators that had tried to understand why you have these variable cell cycle times within these cell populations, they were scratching their heads, and because they had, as in the words of Einstein, there were hidden variables that they didn't know about, the hidden variables. And of course, the hidden variables in our system became unhidden when we were able to determine the concentration of IL-2 and the density of IL-2 receptors. And because of the fact that all cells that have been studied up until this point in time over the last 50 years grew in exactly the same kinds of ways with these log normal rate distribution of cell cycle times, and because of the fact that there was no selection for faster growing cells, we figured that we had really uncovered the molecular mechanisms that were responsible for cell growth that were common to all life forms, everything in bacteria from there on after. So that was good. And we knew at that point in time that the reason that we entitled this paper a new system to study cell growth was because now we knew that we could use our T-cell IL-2 system to delve into the molecular mechanisms that was making these cells, that the IL-2 receptor interaction at the cell surface was causing the cells to move through G1 into S, DNA replication and mitosis, which is fundamental to all life forms. And both normal cell growth as well as abnormal cell growth, meaning cancerous cell growth, which is basically where I started in my scientific career. I started out trying to understand leukemia and quickly realized that if we didn't understand what regulated normal cell growth, we didn't have a hope at all to understand malignant cell growth. But now we had the system to do that. And the very first thing that we did after this series of experiments were published was that we started looking for genes that the IL-2 receptor mechanism turned on. And we had our first paper out, came out in Science in 1986, just a couple of years after this paper. Now, what happened subsequently was interesting to me anyway. So we were all excited. We were turning cartwheels in the lab and so forth, breaking open the champagne and all this stuff. And actually, one of the things that we got when we got we sent it off to Science, we got it back right away with laudatory comments from the reviewers. One guy said, "This is the best paper I've ever reviewed, ever." That's all he had to say. He didn't say anything else. So the thing is, the cell biology people were the ones that were working on this area of science. T-cell people, they didn't care. They didn't even know that there were any questions to be had. And so when this paper came out, because all of the different kinds of investigators are sort of working in their own little silos, the cell biology people didn't pay any attention to this paper. No trumpets went off or rockets flared or anything on this whole thing. And also, at the same time, the immunologists, as I said, they who cares whether or not the cells are variable in their cell cycle progression? And they just were not interested in that. And actually, what they were interested in, and which is what we're going to review next, is they wanted to know what the T-cell receptor did. They didn't care what the IL-2 receptor did. Different strokes for different folks. But as I said, this was really a eureka moment for us because it solidified our hypothesis that the immune system is regulated by a hormonal system meaning that there are hormone molecules, ligands, and there are receptors, and the ligand receptor interaction is what drives the hormone response, whatever it is. And there were all kinds of parameters that had been studied for more than at least 100 years by the physiologists and then the endocrinologists in trying to understand hormone regulation of different cell systems, essentially. So when we tried to sell that idea to the immunologists, nobody really, they didn't care. They didn't want to know about that. And that wasn't in their ken. I like that word, ken. That's the end of that. I admonish you, or I ask you to, before you leave, to like this video and click on the bell and check out the other papers. And don't take my word for it when I will review any of these papers. You really want to go and read them yourself and see whether or not that I'm all full of baloney or whatever, or whether or not you agree with me. And whether there are other papers that you think are even better or worse or whatever. And they're all there on the website. So thanks so much for watching, and I'll see you next time.